Welcome back. And now for our final cases of the day, we will have an immunogenicity case study with Dr. Amanda Lewin. Dr. Lewin is a team lead in the Division of Generic Drug Study Integrity. She was previously a reviewer in the Division of New Drug Study Integrity and planned, conducted, and directed regulatory inspections in support of ANDAs, NDAs, and BLAs. Dr. Lewin completed her PhD in pharmacology at Georgetown University, her MFS in forensic toxicology from the George Washington University, and BS in chemistry from the University of California, Santa Barbara. Welcome, Dr. Lewin.
Good afternoon, everyone, and welcome to the final presentation of this section. I hope you've enjoyed the workshop thus far. I'm Amanda Lewin, and I will be taking us through the immunogenicity case study. As always, here is my disclaimer. This presentation reflects my views and not those of the FDA. As for our learning objectives today, hopefully we will be able to identify potential issues with an immunogenicity study based off of the background provided, understand why those issues may impact sample analysis, and identify what additional data may be available to help evaluate or mitigate these potential issues. So over the next few slides, I will be providing some background on a bioequivalence study with pharmacokinetic endpoints. The study is for a biosimilar product, and an immunogenicity assessment was performed as part of the safety evaluation. I suggest you take a few notes if you are able, since I won't be able to go back to these information slides when we move into our discussion section. So this was Study Perfect 123. It was an open-label, multi-center parallel study of Curamab and AB123 in patients with chronic video call fatigue to assess PK and safety. This was a multiple-course study, though the number of courses differed by subject. So each subject completed at least one course. However, subjects may continue on to additional courses as outlined in a patient's treatment plan, and as long as clinical benefit is observed. So Best Sponsor Incorporated conducted Study Perfect 123 to show bioequivalence between their biosimilar product, AB123, and the reference listed product, Curamab. The study assessed both PK and immunogenicity in a patient population. Samples were sent from each of the clinical sites to a single analytical CRO, DC Analytical Services, that was contracted by the sponsor to perform analysis of both PK and immunogenicity samples. Best Sponsor Incorporated developed a method for the detection of AB123 and Curamab in plasma, and DC Analytical Services was responsible for performing a partial validation for the method transfer. DC Analytical Services was also responsible for the development and validation of a method to detect the presence of anti-AB123 antibodies and anti-Curamab antibodies in plasma. In each course of this study, 6 doses of either Curamab or AB123 are administered over 6 weeks. The PK and immunogenicity samples were collected according to the schedule on this slide. So pre-dose samples for PK were selected during each week, weeks 1 through 6. A 2.5-hour post-dose sample was also collected on weeks 1, 3, 4, and 6. Immunogenicity samples were also collected pre-dose on weeks 1, 4, and 6. There was also a single follow-up visit following the final course of the study where a PK and immunogenicity sample was also collected. So the firm developed a qualitative electrochemiluminescent method for the detection of anti-Curamab and anti-AB123 antibodies in human plasma. The assay validation included the following parameters: screening and confirmatory cut points, sensitivity, precision, specificity, selectivity, drug tolerance, prozone or hook effect, titer precision, and stabilities. So in the rest of this case study, I will present additional information for a few of the assessments listed on this slide. We will then go through the data together and consider what potential issues there are, what additional data we may be able to consider, and what kind of evaluation we can do with that data. Okay, so let's go ahead and get right into the first case set, where we'll discuss the cut point determination. The firm determined the screening cut point utilizing 50 independent normal plasma lots. Each lot was analyzed on 3 separate occasions, overall resulting in 150 ECL values. The site then averaged the 3 ECL values for each lot across the 3 occasions. This resulted in 50 averaged values. It was these average values that underwent cut point calculation, including outlier exclusion. And according to the firm's procedures, they would only undergo up to 3 iterations of normalization and outlier exclusion. Additionally, it was only outliers on the upper bound. No lower bound outliers were excluded. So even if additional outliers were present, if 3 had already been removed, the firm would stop there. So following outlier exclusion, distribution of the data remained non-normal, and thus a non-parametric 95th percentile cut point value was calculated. Ultimately, a correction factor of 1.3 was determined as the plate-specific cut point. The firm determined the confirmatory cut point in a similar manner to the screening cut point. So they used 50 independent normal plasma lots, again assessed on 3 separate occasions. The independent plasma lots were assessed unspiked and spiked with either AB123 or Curamab to determine the percent signal inhibition. And just like with the screening, the average of these 3% inhibition values for each lot was determined, and this is again what was used for the outlier and cut point evaluations. For the confirmatory, no more than 1 outlier, and again only at the upper bound, was to be excluded, regardless of whether there were additional outliers present in the data set. And for the confirmatory assay, the cut point was calculated based on a 0.1% false positive rate. All right, so let's get into the discussion questions for this case set.
All right, good morning, good afternoon, good evening, everyone. Hopefully you can hear me. I presume I'll get notified pretty quickly here if you cannot. So let's get right into the discussion questions for this first case set. So question 1, based on what you have learned thus far in the workshop and the information provided about the study, what concerns might you have with the cut point determinations? So what I'm looking for right here is not necessarily what is wrong, but what might you want to probe a little bit further into. So what I've provided you thus far is really what we would have available to look at as far as method validation data or study reports. But from that data, what might you look into further? So, or ask questions on once you get to a site for an inspection. So I see one comment here that we have targeting a 1% false positive rate. Great, yes, we're looking at 0.1% versus 1%. Great notice. What about, their analyses of the individual lots, the data used to evaluate the cut point? What kind of things might be problematic there? See if we get a few more responses, and I'll kind of go in and discuss what I have thoughts that I have that come up when looking at that kind of data. All right, looking like we definitely caught on to the 0.1 and 1%. All right, so if we go into the discussing this question, the first thing I would point out is that they use the average analysis of the 3 lots. So in their cut point determination, they actually used the 50 average values as opposed to the total 150 individual values. Another point on this, they only analyzed them on 3 occasions. Currently, the guidance does recommend analyzing those individual 50 lots with at least over 6 occasions. So you have at least 2 analysts on at least 3 days. So you had those 300 values to be used in the cut point determination. A second point is they limited the outlier exclusion to only 3 points in the screening assay and 1 point in the confirmatory assay, right? So this is criteria that's not based on the data. It said it's a general practice to limit the outliers removed, regardless of whether there are true outliers that still remain in your data set. Let's see, a third point to point out is they did not exclude outliers on the low end, right? They were only considering outliers on the upper end. Yes, outliers are going to be more present on the upper end. You'd expect to have more, but that doesn't preclude low outliers from being present in your data set and should be evaluated as such. We caught this one great. They used a 0.1% false positive rate instead of our recommended 1% false positive rate. I think you guys get definitely gold stars on noticing that point. This last note, I'm kind of only going to address it in this case and not really much further, but they use normal blank plasma, right? So that's typical in validation. There's nothing necessarily wrong about that. But just to note that the study was done in patients. And so when you do go into a patient population or even a study population, you need to evaluate whether those blank lots used during method validation to establish the cut point, whether that cut point's relevant to your patient population or study population. And it might, and that's fine, but it might not. And then at that point, you would need to move on to determine an in-study cut point. So hopefully you guys got some of those points. So from that, sort of moving on into question 2, how could this method of cut point determination impact sample analysis? So what do these, how do you think this might influence your data when you move into sample analysis? So thinking about using the average of the 3 points versus all of the determinations. What about what happens if they're limiting their outlier removals? What could you see as far as impacting the data there? For the confirmatory, the 1% versus 0.1%, what does it actually mean for your data set? What could that mean as far as false positives, false negatives? Kara Dr. Scheibner in yesterday's presentations really highlighted some of these issues with not having proper cut points set. I'm seeing some more false positives, right? So in which way, right? So we see false positives, that means your cut point might be a little bit too low. So which of these is going to result in a cut point that's too low? Let's see, inconclusive evidence. False negative, right, you have a risk of false negatives with your 0.1 versus 1%. Yeah, so seeing some great answers. So averaging obscure sensitivity. All right, so to try to keep us on time, I'm going to go through into the next slide and discuss some of the points that I think we should bring up. But great job, you guys, on providing some of those answers. So the first thing I would bring up is in relation to averaging the 3 results from the lot. So by averaging those 3 replicates, you're decreasing your data set variability, and that's potentially lowering your cut point. And so it's inflating your screened or confirmed positive samples. The problem with this is that it could result in an inaccurate assessment of immunogenicity because you are actually adding a lot more false positives to your data set. The second point to sort of go over is the outliers, right? So the site only eliminated 3 outliers for the screening cut point and a single outlier for the confirmatory cut point. But the analysis of the data, when you actually go into it, there are multiple outliers on both the high and low end of the data sets and additional outliers beyond that 3 and the 1. So a general point on outliers, though, we do recognize that these are plate-based assays, right? So they are inherently more variable. However, we do want to ensure, right, that the variability included in our cut point determinations is not due to the outliers and instead is representative of the assay variability, right? So if you limit your outlier removal, you risk keeping true outliers within your data set for that cut point determination. And then failure to exclude those could cause the data set to have an elevated variability, right? So this would result in an elevated cut point. Ultimately, this may result in a failure to identify and characterize all of your ADA positive samples, or it could result in false negatives. And everyone wants to stay away from the case where you have false negatives. On the flip side of that, there is a case for too many outliers being removed, and this is something that you would want to monitor. And so if you're removing too many outliers, you're getting to the point where you're potentially removing the assay variability in itself. This is a little bit less common of a scenario, but does happen, and it errs the assay more on the side of false positives in that case. And the final point, the 0.1% to 1%, the 0.1% is more stringent than the 1% false positive rate. So this would result in an underestimation of your ADA confirmed positive. So this is erring on the side of false negatives. And I'm pretty sure someone brought that up in the chat. So really glad that you were able to identify that. So moving right along, we're going to go into the final question for this set, question 3. What data could you review to evaluate the impact of the cut point assessment? So what data, so if you go to the site, you have these questions, what can you look at to see how their cut point assessment impacted the analysis of their study samples? Can you tell if the cut point was adequate or not? What might you look at to kind of interrogate that? Is there any data on site that could be requested and provided? Could the site provide you with anything? As a site, what might you be able to do to justify your data sets? I see research logs, so it's sort of getting back into going at the data. Sorry, I can't go back to the last slide for the moment. There is a downloadable data set or slide set if you can go back to those. Let's see. See how many samples were identified as needing confirmatory. So I think you're pointing out there any additional samples that might need to be moved on. It's a good thought. Looking at known samples, reference standards, controls. Just good thoughts coming in here. And I think, you know, Jessica, I think you had it on the right track looking at research logs, and that, you know, we have all the data. The data's still there. It didn't go anywhere. We can just reevaluate it and see if what the cut point is, utilizing some more recommended approaches, right? So we can look at the original ECL values that are still available. So here I have some sample data. It's only 15 lots. It's not the complete data set. On the left, you can see your screening data. The right is confirmatory. So for the screening, these are raw ECL values. Confirmatory, these are just percent inhibitions. And so what I've highlighted here just represents the outliers identified. And so in the averaged column, that's what the firm would have utilized for their outlier determination. And you can see that there's one outlier for each that was identified as an example. But I've also highlighted within the individual occasion data sets those additional outliers that would have been identified. So yeah, this is just a subset of data, but just wanted to bring it up as a visual so that you can see that there were additional outliers identified and what that raw data set might have looked like with the 3 occasions versus those averaged. So you can recalculate the cut point, right? So the original screening cut point factor was 1.3. If you recalculate the cut point utilizing all 150 data points, but only excluding 3 outliers, results in a cut point factor of 1.5. If you do exclude all of the outliers, the screening cut point factor would be 1.2, 1.21. And so this is an interesting case because you can kind of see that by averaging the data of each lot, the firm compensated in a way for not excluding all outliers. So averaging the values reduced the variability, which sort of compensated for not taking that variability out by not excluding all of the outliers. So you don't necessarily see as big of a jump here as you might have been concerned about. For the confirmatory cut point, the original was 32.4%. So if you recalculate the cut point using all 150 points, but only excluding one outlier, and utilizing that 1% false positive rate, the cut point would have actually been 51.5%. If all outliers are excluded, the cut point at 0.1% false positive rate would have been 26.6%. While using a 1% false positive rate, you get it down to a 20.8% cut point for the confirmatory assay. And this is all fine. You can recalculate that, but ultimately, what's the impact on the data? How does this impact your study sample? So we'd have to go then and review that data as well. Doing that, for the screening assay, implementation of the new cut point, so the 1.21 cut point, resulted in 103 additional study samples that were previously characterized as ADA negative, which one now screen positive, and 2 would require further confirmatory analysis and then potentially on past that. For the confirmatory assay, implementation of the new confirmatory cut point resulted in 72 study samples previously characterized as ADA negative, which now 1 confirm positive and 2 require further testing for the potential presence of neutralizing antibody or potentially onto titer or any other assays that may be in that protocol. So that's 72 out of 330. That's 21.8% of your samples. It's really not a small subset. So this also points out the particular problem with a cut point that's too high and your potential for false negatives. While you can easily recalculate the cut point, you might not be able to mitigate the fact that after sample analysis is complete, there may be no option to continue those samples on to confirmatory, titer, NAB, any other characterization assays. So you risk incomplete data. On the other side, if your cut point's too low, there is also a possibility to recalculate the cut point correctly, and you can eliminate some of those false positives. So I think it just highlights the point of you really want to make sure that your cut point is established properly before moving into sample analysis. All right, so moving right along, we just don't want to get too ahead of ourselves. We can move right back into case set 2. Thank you so much.
Moving along to case set 2, where we will discuss sensitivity and the LPC selection. All right, so here I present the precision data. I do this as it is relevant to the sensitivity assessment in this case. So the firm utilized positive controls of both AB123 and Curamab to assess the precision. This data presented is, let's just say it's AB123 and that Curamab has comparable data. So positive controls were assayed in replicates of 3, each in duplicate in a total of 6 individual runs performed by at least 2 analysts. So here you see values 1A, 1B, 2A, 2B, 3A, and 3B. That's representative of the 3 different preparations and then the duplicate wells. So each value here is a single well value. And you can see them across the 6 different runs. So plates 1 through 3 and 5 through 7 were used for the precision assessments. So the site determined the mean ECL value, standard deviation, and percent CVs for each of the runs and each of the positive controls for intra-assay precision. That data is presented towards the middle of the slide with the light gray column and the dark gray column. The inter-assay precision was also calculated with the mean standard deviation and percent CVs presented on the far right. The MPC was also tested, but it's not shown here just for the space in the slide. The firm utilized the following acceptance criteria. The percent CV should be less than or equal to 25% for each positive control in both the intra and inter-assay precision assessments. Additionally, the global mean of each positive control should be less than the next highest positive control. So the mean ECL value of LPC1 should be below that of LPC2, etc. In this assay, the sensitivity was determined based on the inter and intra-assay precision results, specifically the results for LPC1 and LPC2. The sensitivity of the assay was to be set at the lowest positive control concentration that consistently produced an overall mean ECL value greater than the plate-specific cut point on all occasions and met the inter and intra-assay precision acceptance criteria. LPC1 passed this criteria, and it was the lowest PC positive control evaluated. Therefore, the sensitivity was set at 250 nanograms per mil. Additionally, LPC1 was then used moving forward as the LPC for sample analysis. As an additional justification for the determined sensitivity, the firm noted that there was a false positive rate of 5.7% for the individual lots tested in the cut point and specificity assessments. which is consistent with the recommended 5% false positive rate. They concluded that this suggested that the sensitivity was adequate. Now let's get into case set 2's discussion questions.
All right, let's get right into these. The first one for this one being, based off of what you learned thus far in the workshop, what concerns might you have with the precision assessment, the sensitivity determination, and the LPC selection? So again, like the first one, what are some things that you might want to ask more questions about? Not necessarily wrong, but just that you might want to interrogate further, get a little more explanation from the site as far as why they should or why they chose to do things a certain way. I'm seeing that, yeah, sensitivity assessment should have been evaluated with more PC concentrations, a lot more PC concentrations, right? And yeah, LPC should be set statistically for that 1% failure rate. Right now, we're going to focus on the screening assay just for the sake of time and for this example, so we can, I think from here on out, we could ignore the confirmatory as much as you all might like that assay. I apologize if you care about that one more, but we're going to stick with screening for now. So seeing some good responses. So let's go into discuss this first question. So for the precision, they had a acceptance criteria of 25%. So it was a 25% CV for the acceptance criteria. Just to note that the guidance recommends that 20% criteria. This isn't an end-all be-all. You'd want to go and evaluate the data further, but definitely wanted to point that out as far as the acceptance criteria. For the precision assessments, they also only use 3 independent preparations of positive controls as opposed to the recommended 6. So there were 6 individual data points, but those were all in duplicate. So it was a duplicate of 3 independent preparations. That's about as far as I'm going to go into the precision assessment because it's not the focus here, but I wanted to at least point those out for your awareness. Sensitivity was not done statistically. It was based on the precision data, right? So really there was no assessment of assay sensitivity. It just wasn't performed, and it was based on the fact that the LPC passed precision and was consistently positive. And so just going back to that point again, if you want a little more information on that one, Kara did discuss a little bit more on sensitivity and proper ways of determining that. There's also more information in the guidance for that as well. And finally, you know, the LPC was set before sensitivity was even determined. So it was not based on sensitivity. It was arbitrarily chosen. Maybe the site has development data that they used, but as far as the validation is concerned, it was not based on the performance of the assay. So I think it was Jeff, right? You said based on the sensitivity data, the LPC should be statistically determined to have that 1% failure rate, right? Let's see, there was some extremely high CVs in the run with the plate, right? You know, we'd always go in and look at the data as well. Okay, so let's move on to the third question for this set. Oh, sorry, second, getting way ahead of myself. So how could this method of LPC selection impact sample analysis? So we're going to focus in on the LPC for the remainder of this case study because it's really the important factor here. It's you know, you want to establish sensitivity, so you want to establish your LPC properly based off of that. So why would this be an issue? Why would an arbitrary LPC be an issue? Let's think back to some of the things that Kara covered yesterday. What's the role of the LPC? Why do we even have it? It's not the same as with PK data, right? They're only positive controls. There's no quantitation to these. What is their real function in your immunogenicity assays? And why is it so important that we have an optimal LPC? Yeah, the LPC concentration might be too high. So what's the problem with an LPC that's too high? To skew the data, the LPC data, or are we talking about the subject sample data, maybe down by the cut point? Lower risk for becoming negative. Right, if your LPC concentration is too high, it's not going to be negative. You're not going to meet that 1% failure rate, right? Sensitivity is not accurately representing LPC, right? Sample analysis could be inaccurate, right? All great points. Okay, sorry, we might be getting a bit behind. Let me jump into discussing this, but really appreciate the comments. So yeah, the LPC might not be relevant, and therefore, you know, it wasn't selected based on the assay, so it might not be relevant to the assay. What happens then? Well, your assay is not going to be adequately monitored near the cut point, right? We're very concerned about those false negatives down near the cut point, and your LPC should be set to monitor that. And sort of repeating myself here, but the problem being that you could have false negatives in your screening assay if they're not moved forward onto that confirmatory assay. All right, so moving right along into question 3, I think we're doing great on time. Okay, so how might you assess whether or not the LPC was relevant, right? So they selected the LPC. Okay, they didn't do it perfectly. Okay, that's not necessarily the end all be all here yet, right? What else can we go to see if the LPC was relevant or not? Is there something that might be helpful in how are we going to evaluate this? Failure rates of the assay, great thought. Yep, we can definitely look at that. We have that data for sure. What else might we get? Can we look at anything from study sample? You know, we're on site, we're at the CRO, we can look at anything they have. Any of the data they've generated, we can look for, we can ask for. And we can use that to help us determine the impact on the study data. To enable analysis samples at lower cutoff points. I'm not entirely sure what's meant by that comment. Let's see. All right, so I think we have a few good thoughts there. Let's go ahead and discuss some of this. So yeah, I think it was pointed out that first, okay, sorry, we didn't point this one out. We're going to get to this one. The signals of the subject sample. So you can look at the actual subject data, right? We have all of it. We have the LPCs and the subject sample data. We have all of the subject sample data and the cut point ECLs, so we can look at it. So you can look at the signals of the subject samples versus your LPC and your cut point. And so in the next few slides, I'm going to really hone in on this point, but we're going to sort of pause this for a second. But here is a comment that you guys did pull up, and it's the LPC failure rate, right? So you can look at the LPC failure rate in sample analysis. Did it fail 1% of the time? With smaller studies, this is definitely difficult to evaluate. If you only have 20, 50 runs, you might not have a failed LPC, and that's within the expected response. With the larger studies, though, you can really look at that as well. And then you have false positive rates. So after the study is done, there, you know, you can also look at the false positive rate when you look at those confirmed negative samples. So if the false positive rate is above the recommended 5%, you know, it might indicate that the assay was performing as expected. You know, this isn't a standalone evaluation. I wouldn't necessarily say this alone would verify that your LPC was adequate, but it can provide additional reassuring information. But let's get back to that, looking at the sample data. So your sample data versus your LPC. So in the next few slides, I'm going to show you some graphs. I don't want to ruin the surprise quite yet by going into those slides, but they're going to show you some examples of what we might see. So here's the first example. Here we have subject samples on the x-axis, so just by sample number, and then you have your raw response on the y-axis. Now, I apologize, it's OD, not ECL. I hope you'll all forgive me on that point. But the graph you're seeing, the orange solid line, that's your LPC response, your average LPC response. And then in the orange dotted lines, those are sort of your 20% on either side, looking at, you know, the allowable variation in these assays. And then the cut point would, the average cut point would then be that red line down the bottom. So looking at this, you could get lucky. Maybe this is what happened. Maybe your LPC, even though it wasn't quite selected correctly, might have been perfectly okay. And this looks fine. You know, I would also tell you that in this case, the LPC failed 1.2% of the time in sample analysis, and you had those signals consistently near the cut point. So again, even though the LPC was not selected based on sensitivity, you know, it might have been appropriate in sample analysis. I'll say this is a very rare occasion, so please don't just throw caution in the wind and randomly select your LPCs. This rarely happens. You know, most of the time we're getting an instance that looks like this. So again, your LPC in orange with the 20% in that dotted on either side, and red being your cut point. So in this case, the LPC didn't fail during sample analysis at all. So it was never negative, and it's not monitoring the assay performance near the cut point. So this would not be a suitable LPC. And you do see a lot of samples in that range that's not being really covered by that LPC, especially down near the cut point. So even with this being true, are there ways to assess the potential impact of not monitoring the performance of the assay near the cut point? Could there be other data that we could also look at to help potentially reassure us in this situation? So a few things that come to mind again is you can still look at your false positive rate and see what you're getting there. You can look at the ECL values of your confirmed positive samples. Where are those falling? And you can also actually look at the reproducibility near the cut point. So you have potentially a lot of data here to look at. When samples go through screening, confirmatory, and maybe even also titer, that's actually 3 sets of the same sample being analyzed in the screening assay. And you can look at the results of that and see if your screening assay, especially in those samples near the cut point, is reproducible. And that might be something that can help justify the data set, even though your LPC in this case wasn't so great. So just one more example here. Maybe you get something like this. This does occur every once in a while, but not super common. In this case, you know, your LPC still not relevant, still not monitoring your assay near the cut point, still does not fail 1% of the time. However, you do see a distinct positive-negative population of samples, is what it appears here. And let's also say there was a 7% false positive rate in this assay. You know, could you potentially be a little more, would it be reassuring then that the LPC, even though not relevant, might not have impacted sample analysis? So it's definitely something that you want to be sure your LPC is set correctly. And then if you move into sample analysis and maybe something changes, be sure to monitor it and monitor it in relation to your study samples. Okay, so that's it with that case set. Let's move back into the recorded session with our final case set for the day.
Moving on to the final case set where we will discuss drug tolerance. The firm performed drug tolerance to determine the concentration of drug that could be present in samples without masking the presence of anti-AB123 or anti-Curamab antibodies. Again, let's assume here that the data for both drug products is comparable. So low positive control and high positive control samples were spiked with drug product at concentrations from 800,000 nanograms per mil down to 20 nanograms per mil. Each sample was assayed in duplicate in a single run with both the screening and confirmatory assay on a single plate. So the confirmatory percent inhibition was calculated using the corresponding screening result as shown in this table. So if we look over to the left-hand side's table, and we'll just look at the row for the 800,000 nanograms per mil, this is how much drug is added to each of the wells corresponding to the screening and confirmatory assay. The ECL value for those results are shown in that same row. So the screening assay with 800,000 nanogram per mil of drug on board resulted in an ECL value of 60. For the confirmatory assay, the result was 61. From those two results, the percent inhibition was calculated at minus 1.67%. So for the screening assay, the drug tolerance was determined to be 100,000 nanograms per mil for the HPC and 200 nanograms per mil for the LPC. So you can see here that For the screening assay, the signal did not rise above the plate-specific cut point of 130 until the drug concentration reached 100,000 nanograms per ml. Similarly, for the LPC, the signal did not rise above the plate-specific cut point of 108 until the drug concentration was at 200 nanograms per ml. In the confirmatory assay, drug tolerance was determined to be 25,000 nanogram per ml for the HPC and was less than 20 nanogram per ml for the LPC. So again, on the left-hand table, the percent inhibition did not rise above the cut point of 20.8% until the drug concentrations were down to 25,000 nanogram per ml in a sample for the HPC. For the LPC, the percent inhibition never rose above the 20.8% cut point. Therefore, the drug tolerance was reported as less than 20 nanogram per ml. So even though the drug tolerance was not really established for the low positive control in the confirmatory assay, the firm chose not to evaluate it further and therefore just reported the drug tolerance as less than 20 nanogram per ml. The firm did note that there was a signal suppression by the drug, which was detectable over the range of drug concentrations. However, the suppression of the signal did not affect the ability of the screening or confirmatory assay to detect positive ADA results in the presence of high concentrations of drugs. From that, the firm concluded that sufficient drug tolerance for the method was demonstrated. We can now get into the discussion questions for case set 3.
Okay, so this first question, pretty easy out here. Do you agree with the firm? Do you agree with the firm that sufficient drug tolerance was established? What do you think? I think Jeff has written down here, concentration immunogenicity sample collection. Great way to sort of think along those lines. Yep. Let's see. Drug concentration. Yep. What is the drug concentration? Really focusing. Okay, no's and yeses. See what we're getting. Okay. So surpriser here, maybe. It depends. I guess this is something that you loved hearing from FDA. It depends, but it does depend. And Jeff was getting right on the right track here is what are your concentrations in subject samples? If you know it might not be sufficient, if you know what you're expecting in subject samples, you know that, as Kara pointed out yesterday, you might need to try to improve drug tolerance of your assay. There may be situations in which drug tolerance cannot be improved, but you do need to be aware of those limitations and do the best you can to eliminate that interference if you think it might be an issue in sample analysis. So let's say in this case, the firm didn't know. They didn't know what the anticipated drug concentrations in those immunogenicity samples was. So that's going to move us into question 2. Is there data available to determine if the drug tolerance of the assay was sufficient? Go back to the beginning. Remember, some of the background in this study was, can we look at anything to see if our drug tolerance was sufficient, right? Because just because they established a certain drug tolerance, we don't really have enough information to say yay or nay. But do we? Is there anything else that we can look at? Seems numbers are not like they were expecting. Yeah, I imagine they probably were not very appreciative of that drug tolerance of the assay. I think if we go and think about the study design of this one, it potentially being at certain data points, selection of appropriate sample collection time points. So this brings up a point that don't really have planned to talk to in the discussion, but yeah, when you're thinking about those time points to be collected, you do want to look at what drug on board, what there might be, and whether or not you want to adjust your time points to compensate for potential drug on board. You could look at the guidance, but drug acceptability absorption. Yeah, so here Quite simply, yes. We do have data to look at. I'm going to bring you back to this table, right? So there were PK samples collected at each of the time points that immunogenicity samples were collected. So for every immunogenicity sample, we know exactly how much drug is on board. So we can just look at the data. We go back and we ask the firm to provide your ADA and your PK data together, and we can evaluate that. So we did that, or hypothetically, we did that. So when you combine the data generated from the ADA assay and the PK assay, you can evaluate that drug tolerance. And so in this case, let's say there were a total of 1,500 samples analyzed in the ADA assay. Of these samples, 765 had drug concentrations below the limit of quantitation. So in this case, your PK assay had a 1 nanogram per mil limit of quantitation. 195 samples had concentrations they had drug on board, but below 200 nanogram per mil, which was that drug tolerance established for the LPC in the assay. There were 525 samples, however, that had drug on board above the established drug tolerance of the LPC. This is 35% of the samples in the screening assay. It's not an insignificant amount. And you can parse this down further and look at the percent of samples in each of these three groups that were positive or negative. And so samples with drug concentrations below 200 nanogram per mil, below that drug tolerance, and those with no drug on board had similar percentages in their rates of positivity, negativity. When looking at samples with drug on board above the drug tolerance, there was a slight increase in negative samples. Now, this doesn't mean there absolutely were false negatives that occurred, but it's an indicator that you might have an issue. So another caveat to this data set is that in this first table, samples with drug concentrations below the limit of quantitation were actually from course 1, day 1 pre-dose samples. So these were subjects that had not been dosed with either drug product yet, or they were samples from your end of study follow-up. So this table on the right, this kind of just takes out a subset of data. I'm not going to go exactly into why this subset of data. It's not exactly relevant for this conversation. Just trust me, there's a reason. So this second table includes only samples from course 1, weeks 4 and week 6. And so there are no BLQ samples in this case. So you're just looking at those with drug on board below and above the established drug tolerance. And you see that you're getting at a slightly more differences between those positivity and those negative results, depending on whether or not drug is on board. So let's go to the confirmatory and see the impact there. So for the confirmatory assay, there were a total of 331 samples analyzed. Of these, 199 had drug concentrations below the limit of quantitation of the PK assay, so below that 1 nanogram per mil. 30 samples had concentrations between 1 and 20 nanogram per mil. Remember the 20 nanogram per mil being the lowest drug concentration even evaluated for the LPC in this confirmatory assay. And so there were 103 samples, though, that had concentrations above 20 nanogram per mil. And that's 31%. So you're looking at 40% of your samples with drug on board. And we don't really even have a drug tolerance established in this case necessarily. And so here you can also look at that percent of samples in each of these groups that were positive or negative. And 80% of the samples with drug concentrations below the limit of quantitation were positive. Compare that to 50-50 on your samples with any drug on board. So because of the drug tolerance issues in this assay, there is a concern for those false negative results. And this all would be taken into account by the review division when going in and actually evaluating this data. All right, so I hope you've enjoyed the discussion sections. We just have a little bit more left going back to the recorded slides, and I hope you all enjoy your day. Thank you so much.
I'm going to go through just a couple other notes on drug tolerance to close out this discussion, just sort of things to think about. So the first is optimize your assay. This may sound obvious, but do your best to try to eliminate drug tolerance issues. We recognize that this is not always possible to eliminate drug tolerance as an issue, but if you can get it above concentrations you expect to see, it's really going to be much better as an outcome for sample analysis. If you're not able to establish a high enough drug tolerance, just make sure you're aware of the limitation going in the sample analysis and understand how you might need to compensate or monitor to understand if drug tolerance is impacting your sample analysis. And even if you think drug tolerance was sufficient, it's best practice to even confirm this after sample analysis and looking at your drug on board in your immunogenicity samples. And this consideration, whether or not it was sufficient drug tolerance, can change by study, depending on the dose or the study design. Drug tolerance established in validation may or may not be able to cover all different study types in a specific program. So along that same line, monitoring your drug concentrations during sample analysis becomes important, and important to do that the best you can. We recognize there's limitations, particularly depending on whether your site is responsible for also analyzing corresponding PK samples, or whether you even have corresponding PK samples to know the drug concentrations in your specific immunogenicity samples. Also recognizing you may need to have sponsor cooperation. Ultimately, we just want to, it's best to do what's in your power to ensure that the data you are reporting is being presented as accurately as possible, considering all the limitations in your assay. So it's similar to how you would report samples that exceeded an established stability. That sample might exceed stability that's outside of your control, but if it does go beyond, say, the established long-term stability, either the data tables with the samples will indicate as such and flag those samples as being outside of stability, or they're reported in a different table, but these samples are flagged and noted. So you would similarly do something like that for drug tolerance if available, but ultimately it's about transparency. So being upfront with your data, what limitations it could have and how that might affect the study samples. And all of this is taken into account by the agency when analyzing and evaluating the data that is submitted. So in summary, I'll just go through a quick little note on the key points from this case study. So the first is how the cut point is determined is critical for an assay. Most importantly, it should be established with a scientifically justified approach. The statistics should be used consistently and not changed in the process of determining a cut point. So, for example, using different criteria for determining outliers in the same cut point determination. So maybe using more stringent criteria for your first outlier determination and then subsequently less and less stringent criteria. That would not be advised by the agency. Also, don't forget to evaluate the relevance of a cut point for sample analysis in different populations. So if the method validation used normal plasma or serum, you should evaluate the cut point if the cut point is relevant for a patient population. The LPC of an assay should be statistically determined based on the sensitivity of the assay. It should be set to statistically fail 1% of the time, and the sensitivity should be properly established using a scientifically sound approach. Ideally, drug tolerance should be established to cover concentration observed in subject samples. As I just covered, though, we understand there are limitations. In those cases, the firm should do their best to accurately report the data, including any limitations in the reliability. To finish up, just have this quick little closing thought. Every assay is different, therefore each requires unique considerations, and you should use scientific principles to help guide what considerations may or may not be necessary for your assay or study. And with that, I will finish up here. I want to thank you all for your time, attention, and participation, and I hope you all have a good rest of your day.
Hello, everyone. Sean here again. Big thank you to Dr. Lewin for that presentation. A lot to digest there. I really look forward to going back and going over that presentation in the future. And as that was our last presentation, it's my job to wrap things up. I want to thank you all once again for your time and energy these past 2 days. We know your time is precious, and we're really honored that you chose to spend it with our dedicated staff in OSIS and SBIA. First off, let me thank all of our presenters and our presentation hosts. We could not have done this without all their work. And I really appreciate everyone's patience with some of our technical difficulties, but that's the beauty of these things. There's always new things to learn. I also need to give a big thank you for all of your questions and interactions. I really enjoyed getting your thoughts and know that it will help us do our jobs better in the future. We hope we have provided some useful information about how OSIS supports the drug and therapeutic biologics development and approval process, and that you're now better equipped for your work in this important environment. As the pandemic has made abundantly clear, public health is even more fragile than most of us even imagined. All of your efforts towards bolstering public health and developing critical life-saving and life-enhancing medicines are really vital. I want to thank you all for what you do, and thank you for your time. Hopefully next time we'll be able to see some of you in person. So at this point, stay safe, and I will turn it over to Nora to take us home. Thanks again, everyone.
All right. Thank you.